You're listening to Autumn on the Air, the weekly podcast that brings you conversations about the impact of research commercialization and the people who make it happen. Join us for interviews with patent and licensing professionals, innovators, entrepreneurs, and tech transfer leaders on the issues and trends that matter most. Keep listening for an inside track on the people, IP policies, and politics changing our world.
Welcome to Autumn on the Air. Today, I'm thrilled to introduce our guest, Dr. Yuanbing Jiang, who also goes by Jason, a research assistant professor in the Division of Life Science at the Hong Kong University of Science and Technology. Jason is a rising star in neuroscience research whose pioneering work is transforming how we detect and understand neurodegenerative diseases. Jason obtained his PhD from CUST under the supervision of Professor Nancy IP and is a junior member of the Hong Kong Research Grants Council Research Fellow Alliance. His research focuses on developing diagnostic and therapeutic strategies for neurodegenerative diseases using proteomics and genomics. He has established integrated high-throughput multi-omics data sets, including the world's first comprehensive blood proteome profiling for Alzheimer's disease. This foundational work laid the groundwork for the development of the first multi-protein blood biomarker test for Alzheimer's disease, for which Jason is the key inventor. The technology has been licensed to Cognitect. His research aims to identify novel biomarkers for neurological disorders, enabling early screening, staging, and monitoring of these conditions. Additionally, Jason integrates large-scale multi-omics data analysis in human subjects with systematic experimental validation in animal models, seeking to uncover potential pathogenic factors and mechanisms underlying neurodegenerative diseases and contribute to the development of innovative therapeutic strategies. Today, we'll discuss Jason's groundbreaking work on blood-based biomarkers for Alzheimer's disease, a simple blood test that's achieving 96% accuracy in detecting early-stage Alzheimer's and has already benefited over 1,000 patients in Hong Kong within 6 months of its 2025 launch. This innovation represents a potential paradigm shift in how we diagnose and intervene in Alzheimer's disease worldwide. Welcome to the podcast, Jason. Really excited to have you on the air today.
Very glad to be here. Nice to meet you, Lisa.
Yes, very nice to meet you as well. And I thought, Jason, the best way to start the podcast is to ask you if you could tell us a little bit about the journey from conducting the world's first large-scale proteomics study of Alzheimer's disease to identifying the 21 protein diagnostic panel. Tell us how that journey unfolded.
Yeah, sure. So we start with journey with the very critical clinical needs. So at that time, the Alzheimer's disease is a very serious and important challenge to the field. So this disease is not like other traditional other disease kinds of diseases. So it has the pathological changes 10 to 20 years before they really have the clinical symptoms. And due to this, patients usually only come to the hospitals to see the doctors when they have the obvious clinical symptoms, which already becomes the late stage of the disease, and they meet the golden window for the treatment. And at that time, there are some effective diagnostic tools, like the amyloid PET imaging or the cerebral spinal fluid measurement of the biomarkers. But those methods are either very expensive or invasive that cannot be really applied to the clinical settings in a large scale. So that's why we are thinking about whether we should develop a blood test, which is much more convenient and easier and faster and can be used to almost everybody. By that time, although there are some biomarkers already be identified by previous studies, but they are not good enough. They don't have very high performance due to the limitation of the traditional detection technology. like the ELISA method. They are not sensitive enough. That's why we are thinking if there are new methods, we can detect more biomarkers and more sensitive, we can achieve that. And the turning point is the emergence of the ultra-sensitive and high-throughput proteomic assays. So there comes to new biomarker assay detection method, and that can measure more than 1,000 blood proteins with even 100 times higher sensitivity. And this exactly aligns with the characteristics of the Alzheimer's disease, because Alzheimer's disease is not just a brain disease. Actually, it involves the dysfunction of many aspects of the body system, like the immune system or the metabolic system. And when we use these new methods to apply to the blood system of the AD, we can cover different aspects. That's why we can measure different biomarkers. And based on this, we conduct the first comprehensive assessment of the blood protein of Alzheimer's disease. And we measure 1,000 blood proteins to assess the activities of different aspects of the organs, the tissues, the biological processes of the AD patients. And we identify more than 400 new blood biomarkers for Alzheimer's disease. And that's why we select some of those key biomarkers that to form a 21 protein panel to cover the different aspects of the disease processes and to achieve the very accurate detection and assessment of the disease.
So, Jason, given what you just told us about the assay, how does it provide biological annotations for different stages of Alzheimer's disease? And why is this capability to monitor disease progression so important for therapeutic development?
Right. So one of the key insights from our proteome studies in AD is that we find there are many biomarkers dysregulated in the AD blood. And those biomarkers are actually from different biological processes, and they actually have different dysregulation patterns. For example, the dysregulation of the immune system actually it happens at the early stage of the disease when the patients actually have no obvious clinical symptoms. While the neurodegeneration and the death of the neurons in the brain, it usually happens at a later stage when the patients already have very obvious the clinical decline or the clinical impairment. And that becomes the basis of our biological annotation or the staging of the disease. We can use those different biomarkers at different stages to help people to stage the disease. And that is very important. We are not just telling people whether they are having the disease, but actually we are telling people that what kinds of the disease they have and at what stage. And it's critical for the treatment because some of the drugs they can only treat the patients effectively at a specific stage. For example, the anti-amyloid drug, they only treat the patients at early stage when the people have mild cognitive impairment and mild AD, and which is not effective at the later stage. That's why we need to use such kind of blood tests to identify those people at early stage to allow them to have effective and timely treatment.
Now, the 21 protein assay achieved 96% accuracy in detecting early stage Alzheimer's, which outperformed plasma ATM biomarkers. What technical innovations enabled this level of performance, and why does accuracy matter so much in a screening context?
Right, so we will say there are two types of key innovation. One is the assay detection technology, like I said. So the emergence of new assay technology by detecting 100-fold higher sensitivity and more blood proteins at the same time, this really changed the field that we can allow us to have the measurement of those biomarkers that cannot be measured ever before. So that is one of the key innovation. The second one is the concept. So previous studies, they only focus on the ATN biomarkers. And they believe that the ATN is the key is the whole picture of the disease. But at that time, we proposed a new concept that we should not only focus on ATN, but we should focus on the whole picture of the disease that also includes the immune system or the metabolic or vascular. That is also another key innovation that by measuring those different aspects of the diseases, then we can achieve a higher accuracy and more comprehensive assessment. And that accuracy is super important because that can really reduce the chance of misdiagnosis, which is very important in the clinical settings because the misdiagnosis it can really cause troubles. The patients will suffer a lot when they have such kind of conditions. And also it can help us to achieve the disease earlier when we detect the disease. And that is also due to the higher accuracy.
Now, you validated the biomarker panel in both Hong Kong Chinese cohorts and a Spanish biodegmar cohort. Why was it critical to demonstrate accuracy across different ethnic populations? And what do you think this means for global applicability?
Yeah, because we believe clinical use for blood tests must have the robust performance across different ethnic groups. That is very important too, and also very essential for the application of such kind of blood tests globally. And it is also important to show that such a diagnostic test is actually capturing the core disease-related features, not due to the background noises of the specific cohort. And this really can help strengthen the confidence of such kind of blood test. And we are very encouraged that our assay really show good performance across different ethnic groups that really can demonstrate that such blood tests can have the potential to be applied to different people, different regions, and different countries.
So, Jason, I know the blood test reduces diagnostic costs by up to 88% compared to PET imaging. Tell us, what does this mean for healthcare systems and patient access, particularly in resource-limited settings?
Right. So in the resource-limited regions, reducing costs is the very important factor because the PET is super expensive. And in those regions, most of the patients don't have the chance to really access to such kind of high-tech and super expensive technologies. And as a consequence, like I said, they only go to the hospital to get a diagnosis of disease when they already develop very obvious symptoms or the cognitive decline or the memory loss. And they already at the late stage of the disease, they miss the window for the effective treatment. And eventually the whole journey of their patient care is very horrible. And the blood test is much simpler than the amyloid PET. It's cheaper, it's faster, and that can be accessible by almost everybody. And that really can accelerate the diagnosis of the disease and to allow the patients have the timely treatment or the intervention of the disease. And that is super important in the clinical settings.
So, Jason, how does having a simple blood test for early Alzheimer's detection change the pathway from initial concern about memory to diagnosis and intervention? And what can patient families now do with an early diagnosis that wasn't possible before?
Right. So a blood test is more, it can achieve the diagnosis much earlier, faster, and more accessible. And that is key to the diagnosis of the disease. For patients, like I said, they can have the chance now to get a timely treatment like the anti-amyloid drugs when they get the early diagnosis of the disease. And for patient families and caregivers, they also have the chance to have the proactive care planning. That is also very important because they can have more time to organize the support and discuss the future care preferences and address the daily needs. That is also important to save the time and the resources during the caring of the patients. So generally, so for such kind of early diagnosis, it's not only providing an opportunity to know the disease earlier, but more importantly, to provide a chance for the patients and their caregivers a chance to act earlier for the disease.
So I know these biomarkers can accelerate recruitment into Alzheimer drug trials. With new anti-amyloid therapies gaining approval, how critical is early biomarker-based identification for ensuring patient access to these treatments?
Well, so with the emergence of the anti-amyloid drugs, now the early biomarker-based detection is not an optional option. Actually, now is the key for the diagnosis of the disease, because such kind of a drug requires the confirmation of the amyloid or other biomarker presence in the brain. So that makes the early biomarker-based detection become one of the most important steps in the diagnostic journey of the patients. And the blood-based biomarker can accelerate this to let people have earlier chance to have the detection of those biomarkers. And also it's important for the clinical trials to have such kind of blood-based biomarker detection, because previously when the drug companies, those pharmaceutical companies, to identify patients, they don't know what patients they should look at. And then that they spend a lot of time and much money to find the suitable patients. But now when they have the blood biomarker test, they can screen a large proportion of the population to quickly identify those patients. And that really saves the resources for them to let them to focus more on the research and the drug development. Then that will accelerate the clinical trials and the development of new drugs.
So, Jason, what are the key next steps for validating PlasmaMark AD in broader populations? And what's your timeline for global expansion, particularly into public health sectors?
Right. So the key step for the next phase is the validation of our blood test in more populations and more regions, and to get the regulation approval. That is the essential step for the real application of our blood test into the clinical settings. And we are hardly working on this, because now we are getting the support by the Hong Kong government, and we got the funding support from the RAISE Plus scheme to allow us to have the validation and a clinical trial of our blood test in Hong Kong and in mainland China, and also at international regions and countries like in the Europe or US. So we are collaborating with different clinicians to have a very large scale validation and of this blood test and collect the clinical data to allow us to apply for the regulatory approval at those local regions, and eventually to allow us to get into the hospital to provide such kind of blood test to the patients.
What gives you the most hope about our ability to change the trajectory of Alzheimer's disease over the next decade?
So based on our recent findings, we have already identified so many new biomarkers, and we demonstrate we can develop them into a blood test to detect and diagnose the disease earlier. And I'm very encouraged by such kind of findings, not only for the diagnosis, but also for the future therapeutic developments that I said they can accelerate the development of new drugs, not only the anti-amyloid drugs, but focus on other biomarkers and to have more effective treatment of the disease. And we can also have even the prediction of the disease risk before they really develop disease. And then we can have the timely intervention of the disease risk, so that we call the prognosis of the disease. And they all can be integrated by the AI. Like the AI is a very powerful tool, and that can further improve the performance of the diagnosis, the prognosis, and also the drug development. And together, those can reshape the future of the disease whole picture. And I definitely believe that in the future we can really treat the disease effectively and we can cure those diseases.
Well, Jason, congratulations to you and your colleagues on this amazing achievement with this 21 protein biomarker panel. Not only is it an incredible scientific breakthrough, but it really shows us how academic excellence, strategic tech transfer, and entrepreneurial innovation can really rise to the occasion to meet some of the world's most pressing health challenges. Congratulations again.
Thank you very much.
Jason, thank you for sharing your extraordinary work with us today. Your journey from building the world's first comprehensive blood proteome profiling for Alzheimer's disease to developing a blood test that's already changing lives for patients is truly inspiring. The 21 protein biomarker panel represents not just a scientific breakthrough, but a demonstration of how academic excellence, strategic technology transfer, and entrepreneurial innovation can converge to address one of humanity's greatest health challenges. The impact of this work extends far beyond the laboratory. With 55 million people worldwide living with Alzheimer's disease, and that number projected to rise dramatically as populations age, accessible and accurate early detection tools are becoming increasingly critical. Plasmark AD's 96% accuracy, combined with its cost-effectiveness and minimal invasiveness, offers genuine hope for timely intervention and better patient outcomes. As Cognitech expands globally and continues validation studies, and as Jason and his colleagues at CUST advance therapeutic discoveries alongside diagnostic innovations, we're witnessing the emergence of a new era in Alzheimer's care, one where early detection enables early action. For our listeners interested in learning more about Jason's work, you can visit the Hong Kong Center for Neurodegenerative Diseases at hkcend.org and follow developments from Conitect Limited as they work to make this transformative blood test available to public health sectors worldwide.
Thanks for listening to Autumn on the Air with Lisa Mueller. Get social with us and share your thoughts. You can tweet us at AUTM or visit us online at autm.net. We'll be back next week on the air. Be sure to join us.